There we go. Right, can everybody see that?
Yep.
Everybody can see it and you can still hear me. Lovely. So I suppose I ought to start with a little bit about me, since I'm not completely certain that everyone in here knows me already. So Matthew and Steph probably recognise this lovely glass-fronted building down Cathedral Street in Glasgow. I'm currently in my first year of a PhD in Strathclyde Institute of Pharmacy and Biomedical Sciences at the University of Strathclyde. I'm not actually in this pretty glass-fronted building. I'm in the black and red one on the left-hand side of this photograph. I'm also split between SIBS and the Department of Biomedical Engineering, and exactly why this is the case will become apparent later on. I've also not always been a biomedical scientist. Both of my previous degrees have been in chemistry. So I read for my master's degree in chemistry under the supervision of Professor David Smith at the University of York. Hang on, I think I've gone too many slides there. Hang on, there we go. Right. So, now you know who I am. I suppose we should get started with the science. Firstly, what is nanomedicine? So nanomedicine is defined as being the area of science which studies the application of nanotechnology to various clinical uses. These include diagnostics, aiding the detection of disease, medical imaging, which is frequently new contrast agents, and therapeutics for the treatment of disease. The National Nanotechnology Initiative in the US defines nanotechnology as being the study of phenomena which occur with objects between 1 and 100 nanometers in size. But this upper size limit is considered by many to be somewhat arbitrary, and many examples where strange phenomena occur well above this size threshold can be found. These include the enhanced circulation times of liposomes up to 200 nanometers in size, and the enhanced accumulation of nanoparticles in tumours up to 400 nanometers in diameter. So why are nanomedicines so useful? There are many reasons why nanomedicines are used clinically. In recent years, with the advancements in omics technologies, we're seeing more and more new biological targets being uncovered, and with this, very creative chemistries to treat disease. With new chemistries comes an entirely new set of challenges. There's also a balance that needs to be struck between efficacy and toxicity of any new compound. You don't need to have taken them to know that chemotherapeutics come with an extensive list of side effects. Many of these come about because of the systemic distribution of the drug throughout the body. Nanoparticles are particularly useful here because the distribution of the drug payload can be much more targeted to the site of disease or tissue of interest, reducing much of the accumulation in other unwanted tissues, therefore reducing the severity of side effects and frequently increasing the drug's therapeutic index too. Furthermore, nanoparticles can be used to overcome a variety of issues with the free drug itself. Many novel cytotoxic drugs are effectively like brick dust and are really difficult to get them into solution. So what we can do is incorporate and encapsulate this drug into a nanoparticle, increasing its solubility. Attaching the drug to or into the nanoparticle also imparts further interesting properties, such as reducing the ability of the target cell to remove the drug, known as efflux, and can also contribute to the reduction of multi-drug resistance in both cancer cells and bacteria. Nanomedicines are often considered to be a golden bullet for treating disease, as cell-specific targeting is possible. Nanomedicines have also had a long history of obtaining clinical approval, the earliest example of this being Doxil, a liposomal formulation of doxorubicin, and was given clinical approval in 1995 by the FDA for the treatment of corpuscular sarcoma in HIV-positive patients. Currently, there's approximately 50 examples with clinical approval, but more than 100 clinical trials involving nanomedicine currently ongoing. What this painfully slow increase in the number of nanodrug formulations actually shows us is that there are clearly difficulties in the translation of these promising drugs from lab bench to patient bedside. And finally, there's been a rapid expansion in the market for nanomedicines, worth less than $40 million in 2006, 100 million by 2014, and the most recent predictions as of March 2020 is that it will be worth greater than $1.7 trillion by 2023, although it will now be interesting to see what impact COVID-19 actually has on this. As has been mentioned previously, there are approximately 100 clinical trials involving nanomedicines that are currently ongoing, but very few drugs ever get clinical approval, so clearly something is going wrong. It is known that there are several challenges in getting these novel drugs from bench to bedside, and these issues can broadly be classified into three groups. Issues with manufacture, so difficulty with obtaining batch-to-batch consistency, difficulties with characterising what you've actually produced, and some of these issues I will show you later. Biological fate, so what the drug actually does and how it behaves once administered to a human body. One of the biggest issues, and the one with the most industrial interest, is understanding the developing protein corona and how that impacts on the biological fate of a nanoparticle. As things currently stand, there is no universal opinion as to what parameters are fundamental for understanding the biological fate. And what I don't want you to do here is actually try to read every single bubble. What I just wanted to show was the wide range of parameters that impact on the ability of a novel nanoparticle to obtain regulatory approval and enter clinical use. So most nanomedicines are developed as parenteral formulations, that is, they're given intravenously and they quickly enter the bloodstream. Almost instantly does the nanoparticle then develop an outer coating of proteins and other biomolecules. Depending on where you look in the literature, this is either termed a protein corona or biomolecular corona, the latter recognizing that this coating doesn't just contain proteins. However, the other biomolecules aren't of interest here, and I will use solely the term protein corona. So this protein corona is known to impact the biological and chemical identity of the nanoparticle, but also impacts on how well the immune system is capable of identifying these nanoparticles as being non-self. And the protein corona consists of many different parts. We have the previously naked nanoparticle in the center. Then there is the hard corona. These are the proteins with the highest affinity to the nanoparticle and such are the tightest bound. The soft corona then comprises of proteins which have a much lower affinity to the nanoparticle and are much more loosely bound. Characterization of the protein corona is then difficult for many different reasons. If you want to characterize the soft, loosely bound corona, you'd first want to remove any excess unbound protein. And the easiest way to do this would be simply to centrifuge the nanoparticles and resuspend the pellet. But by doing this, you also throw off this corona in the process. So maybe we should analyze the hard corona instead. It's much more strongly bound, so maybe this will be really easy, right? Wrong. The hard corona is nice and stable if and only if the environment doesn't change. Change the environment and you end up changing the composition of the hard corona. Can anyone begin to see why it's the protein corona that can be such a bottleneck in the development of novel drugs and is actually really irritating? So how can we actually analyze the protein corona? Analysis methods will vary depending on what specific parameters are of interest, but broadly these can be split into two distinct groups. Direct methods such as SDS-PAGE and protein assays, or so-called indirect methods such as nanoparticle tracking analysis and dynamic light scattering. And what advantage that these indirect methods have over the others is that the removal of excess unbound protein isn't actually required, and the nanoparticle corona, then the corona that develops around the nanoparticle can simply be measured in situ. If we go back to the mind map I showed you earlier, the sections that I'm currently focusing on are the material of the nanoparticle, its surface coating, its size, and various incubation parameters, including the protein source, incubation time, and temperature. And eventually, we will also look at cell culture methods, and this is why I'm split between SIBS and biomedical engineering, as we're looking at developing microfluidic models. So the nanoparticles that I'm currently working with are PLGA, usually known as either polylactic co-glycolic acid or polylactide co-glycolide. We're particularly using this one because of its long history of clinical use and the absence of side effects from using it. The additional absence of the polyethylene coating enables us to modify the developing protein corona. And furthermore, if we alter the ratio of lactic and glycolic acids in this polymer, we can also start to modify some of the behaviours of the polymer itself. So what have I actually done so far? So we've taken PLGA and PLGA-PEG nanoparticles, incubated them in human or fetal bovine serum for between 0 and 24 hours at either 25 or 37 degrees Celsius, allowed them to develop a protein corona, and then gone on to analyse them using dynamic light scattering, nanoparticle tracking analysis, and resonant mass measurement. Since as I've made the assumption that most of you are not analytical scientists, I'll now walk you through how all three of my analytical techniques actually work. So dynamic light scattering relies on light being shone through the sample, and the particles within the sample scattering this light. It also relies on one significant assumption, and it does this. It relies on one significant assumption that the particles which are doing the scattering are spherical. And what this assumption then does is it allows the system to fit the scattering pattern to the Stokes-Einstein equation and then calculate the size of the particles that are in the sample that have done the scattering. And nanoparticle tracking analysis functions on much the same principle, but that's where the similarities stop. So nanoparticle tracking analysis shines light through the sample, but what it also does is record video footage. If this decides to play, we'll get a 10-second snapshot of the footage that we get, and I don't think it will. So just imagine that you can see these white dots here moving across the screen. The computer records this footage and later uses it to simultaneously identify the centre of and then track frame by frame the movement of each individual particle. And this is something that dynamic light scattering cannot do, being in an ensemble method. And then again, the system will fit this new data to the Stokes-Einstein equation to give the size of the particles that have been detected within the sample. And finally, resonant mass measurement. This simultaneously occupies the position of my favourite and least favourite analytical method. I currently miss it, but post-COVID-19, I'll be quite quickly back to calling it names and threatening to throw it out of the window. So for the Archimedes system, it contains a microfluidic chip. Inside this microfluidic chip is a microfluidic channel containing a resonating cantilever. This cantilever then resonates at a specific frequency and will continue to do so if not disturbed. If particles pass by the cantilever, the resonant frequency changes, and the bigger the change, the bigger the particle that has just passed by the cantilever must be. The system then uses this change in frequency to calculate the mass of the particle. And since we've already told the computer how dense the particles that it's looking for are, it can then calculate particle diameter. It is also able to calculate particle concentration, solution viscosity, and if I really wanted to, I could simultaneously analyse samples containing both positively and negatively buoyant particles. And now here we get onto the fun bit, and it's here where I get to talk about the first set of data they've collected in the lab. So this first slide shows us what the nanoparticles look like without the protein corona. These are effectively shot-bought nanoparticles, and we can see that they are both approximately 100 nanometers in size via DLS. And in-house, we've also confirmed this size by nanoparticle tracking analysis too. And they've both got really narrow size distributions and are both moderately negatively charged. And this is known to have an impact on the specific composition of the protein corona that will develop, but we haven't got quite as far as this yet. So at first, we're looking for an in situ method that allows us to measure the change in particle size without needing to recover the nanoparticles first. So we've analysed by dynamic light scattering solutions of nanoparticles in various concentrations of fetal bovine serum. And what we can see between neat serum and a 1 in 10 dilution, that actually very little changes. We start to see something here but it's not until we get to a really, really low concentration of fetal bovine serum that actually we resolve the two clear peaks that we wanted. But this particular concentration of fetal bovine serum is so far removed from physiological conditions that any data that we do get isn't really biologically relevant. And we see much the same thing if we then choose to look at the volume distribution of these same samples. So next, we went on to use nanoparticle tracking analysis to look at how the PLGA nanoparticles behave when we incubate them in phosphate buffered saline. This is essentially salt water with additional phosphate, and it's quite nice and boring. So what we see here is a distribution shift over time. More and more larger agglomerates are formed, but what this doesn't show is the formation of a protein corona. It's impossible because there's no protein in phosphate buffered saline. And overall particle concentration decreases irrespective of surface coating or incubation time and temperature. And this effect is most pronounced at physiological conditions, which isn't particularly surprising. And we see much the same thing when we incubate the PLGA and PLGA peg nanoparticles in fetal bovine serum. So why does the particle concentration actually nosedive like this? So this happens because each sample contains a fixed number of nanoparticles within the sample. As these particles agglomerate, they'll form a smaller number of larger sized particles. So with the image here, each frame is supposed to represent 20 particles. At the left hand, this is what the sample looks like just after I've mixed it. And then on the right hand side, this is effectively supposed to demonstrate what we start to see after 24 hours of incubation time. So as we've said previously, we don't particularly like dynamic light scattering as a technique for doing this. But why do we have a preference for nanoparticle tracking and analysis and resonant mass measurement? Here we can see with the pink bar the size of unbound protein in aggregates, but also the size limits of resonant mass measurement, nanoparticle tracking analysis, and DLS. And what we can see is how so much of the range that dynamic light scattering uses is dominated by protein, so that the DLS effectively gets confused. But surely the same thing happens with NTA. The size limit at the bottom end is the same. And it looks like that, but what you're able to do with nanoparticle tracking analysis is tell it what are the things you're interested in, so in this case the PLGA nanoparticles, and tell it to effectively ignore anything else, so that would be all the unbound protein. And resonant mass measurements, on the other hand, with the settings that we particularly use, the bottom end of its size limit is almost set so high that it can't even see any monomeric proteins. What we can then do is apply both of these two techniques together to analyse samples. And the greyed out area in these particular graphs is the size range of data that we can get from the nanoparticle tracking analysis that the resonant mass measurement effectively cannot see. And what this allows us to do is determine that a lot of the stuff that we saw sub 100 nanometers by NTA is definitely protein, and other smaller aggregates and isn't necessarily what we're interested in here. If we then look at the shape of the data that we get from NTA and that from resonant mass, we can see that the shapes map each other fairly well. And that strong agreement is quite a good thing to see between these two techniques because they operate in two different ways. So nanoparticle tracking analysis is an optical technique, whilst resonant mass is not. And so why is doing this even interesting? So as far as we're aware, this is the first time that both nanoparticle tracking analysis and resonant mass measurement have been applied to probe the protein corona that forms around PLGA and PLGA peg nanoparticles. And the more data that we have on how nanoparticles behave on being incubated in serum, instead of going, here is my nanoparticle, how does it behave? Eventually we'll be able to go, I want my nanoparticle to behave like this. So what does it need to look like? So essentially reverse engineer the nanoparticles for clinical applications. And then we are more likely to develop nanomedicines which gain clinical approval. So where do we go to next? So it'd be interesting to undertake total protein quantification so that we can see how incubation conditions, so time, temperature, and the choice of serum affect the composition of the protein corona. We've also run the hardest size to analyse already, and we're hoping to run larger nanoparticles once we're able to get back into the lab, and hopefully this analysis will also be much easier to do. We're also hoping to run proteomics analysis at Glasgow Polyomics facility, but rather than looking the most abundant proteins, which is generally what is done with proteomics analysis of the protein corona, we're actually more interested in the proteins which have the biggest influence on the biological fate of nanoparticles. We will also develop microfluidic models to better mimic the biological conditions such as circulation time and shear rates that the nanoparticles will be exposed to in the bloodstream, and then go on to evaluate binding affinity and kinetics of the proteins that we have found via proteomics analysis. So in summary, we're able to show that particle size distribution varies as a function of serum concentration, incubation time, and temperature, that dynamic light scattering is not a particularly suitable method for in situ monitoring of changes in particle characteristics at physiologically relevant protein concentrations, and we do indeed see and can demonstrate an overlap between the size distributions that we see for our particles via NTA and resonant mass measurement. So there are a lot of people who I need to thank, and not all of these are actually in this photo. So there's quite a few people who are in our lab as part of Philip Sieb's group who are certainly not in that photo, who have been massively supportive over the past few months. I also need to thank the Scottish University's Life Sciences Alliance, as they've given us quite a lot of funding to undertake the proteomics analysis that we were hoping to do this summer. Our Erasmus student Merit, her Erasmus internship was funded by the European Commission, and we're really grateful to Melvin, as we've had quite a lot of contact with Elise, Liam and Dr. Yusuf Chen to help us with not only interpreting the data that we did get from the nanoparticle tracking analysis, but they also really kindly lent us the kit for us to actually do the work as well. That is me.